Now I invite Fiorela to start to show her screen, to share her screen. Okay. Fiorela, are you there? How are you? Hola, Fiorela.
Hello,
so now we have...
do you see my screen?
Yes, we can see it. Can you change the screen, the slides? Okay, perfect. So now for concluding this session, the last session of our meeting, we have Fiorela Alassia and Mariana Córdoba with this paper entitled A Processual Ontology Approach in Biochemistry and the Review of the Structure-Activity-Relationships Dogma, the case of the drug receptors. I will tell you when you have 5 minutes left. And if you have any question, just put a Q on the chat. So welcome, Fiorela.
Okay. Mariana.
Hello. Hi, everyone. We are and myself, Mariana Córdoba, from Argentina. And today we will present a processual ontology approach in biochemistry and the review of the structure-activity-relationship dogma, the case of drug receptors. I will stop my video because my internet is not very well. I will ask you if you can't listen to me, please tell me. So I repeat. Okay, thank you. We are presenting a work in progress from Fiorela's doctoral project. We will analyze the case of drug receptors and biosignaling under the consideration of a process ontology approach. We will try to answer whether this ontology can be applied to the case study and if this allows us to review a central dogma in biochemistry, the relationship between the chemical structure and biological activity. In order to do so, we will briefly mention some basic ideas of the structure-activity relationship paradigm and some challenging cases. Secondly, we will introduce the case of drug receptors and biosignaling. We will refer to general aspects of process ontology, and finally, we will try to answer whether this ontological approach can be useful for our study case, and we will present some concluding remarks. So let's start with our first objective. A central paradigm in biosciences is the deterministic and unidirectional relationship in the chemical structure and the biological activity of a molecule. In the field of protein studies, it is reformulated as the deterministic relationship between the primary structure, amino acid sequence, the protein three-dimensional structure, and the protein function, as follows: sequence, structure, function. This represents the core paradigm within numerous kinds of methods in predicting protein structures, for instance, homology modeling. Here, we can see the hierarchical structure of proteins by a well-known molecular biology manual by Lodish. Amino acids are linked together by peptide bonds into linear chains that can locally fold to acquire a secondary structure. The next level of hierarchy is achieved by a complex developmental folding process in which the protein acquires a specific three-dimensional structure. The functional structure in a relevant environment is called native structure by biochemists. We can find some specific cases that challenges the sequence structure function paradigm, such as moonlight proteins, which have different functions according to their actual environment context, as we can see in this figure, also the intrinsically disordered proteins that may challenge the sequence structure function paradigm since they have unstructured regions in the primary sequence. Epigenetics may be an additional factor influencing the final protein shape.
Okay, so let's move on to our study case, receptors and signaling. Well, receptors are proteins found in cells that act as transmitters of internal or external chemical signals, for example, a hormone or a drug, which may trigger a certain biological response. They are usually located in the cell membrane or inside the cell, as we see in the figure. And the signal, also called ligand, binds to the receptor in a highly specific way, usually referred as to the key lock analogy. Here we have another figure that represents the interaction between a receptor or interaction points between a receptor, in this case, beta adrenergic receptor, and an agonist drug, which is salbutamol, an anti-asthmatic drug. Okay, here we see the insulin receptor signaling mechanism, which promotes, in one case, cell growth and differentiation, but also glucose uptake from extracellular space into cytoplasma. Well, let's move on to the process ontology point.
Okay, now I follow. Let's see now how can a process ontology be characterized. According to the substantial ontology, substantial ontology is the idea that the world is populated by things, by substances in the philosophical traditional sense. According to this view, things have spatial extension. They are stable objects in time, which means that things are objects that persist in time. Their nature and behavior do not depend on external relations with the environment. They are subjects of change. They do activities. Things are, according to this ontology, the fundamental entities of the world. On the other hand, when we have a process ontology, I mean a world of processes, processes are has temporarily extension. They depend on change for their existence, which means that change is more fundamental than persistence when we think of processes. Their nature and behavior depend on relationships with the environment. They are relational entities. It is possible to individualize them, and processes are the fundamental entities of the world. The classic antecedents of processual ontology include the work of Heraclitus, Whitehead, more recently Rechter, Cobbs, and Seibt, among others. Due to lack of time, we will not describe their ontological proposals, but we will see in more detail the work of some philosophers of chemistry and biology that focused on the processes and dynamic aspects within these sciences. In chemistry, we have the ideas of Schummer, who claims for the relational aspects of the chemical knowledge. He claims, chemistry at the core is the science of peer relations. Instead of studying isolated objects to be measured, compared, and put into a classificatory scheme, dynamic relations between objects constitute the basic set of chemical knowledge, and at the same time provide the grounds for the classifications of the objects themselves. Paul Needham, in his work, we can appreciate the use of process ontology for an interpretation of certain concepts of macroscopic chemistry. the physical interpretation of the macroscopic view represented by thermodynamics is naturally understood to involve processes alongside continuants in the fundamental ontology in such a way that their properties are intimately related. We have also the work of Stein, who proposes a strong processism based on Cobb's ecological model of molecules. Based on new models of enzyme function, Stein claims that enzymes are ecosystems, relational interconnected complexes. His central idea is this: the spatial three-dimensional disposition of active site amino acid residues about the substrate is clearly of critical importance for the enzyme chemistry, but also it is the protein matrix in dynamic communication with these residues that imparts to them the status of an enzyme catalyst. Furthermore, these active site dynamics are ultimately coupled to the energy bath of bulk solvent. This brings us to a new understanding of the origins of the catalytic power of the enzyme. To pay the energetic cost of catalysis, the enzyme protein transduces energy that is available from its stochastic interactions with the medium to the active site. We also have the works of John Dupré and Daniel Nicholson. According to them, the building blocks of living reality are not individual things, but processes. Things are abstractions of stable processes. They are precipitates of processes. The living world is a hierarchy of processes, stabilized and actively maintained at different time scales. We can think of this hierarchy in broadly mereological terms: molecules, cells, organs, organisms, populations, and so on. In the same line, we have Guttinger work. Guttinger claims that macromolecules like proteins or DNA are relational entities that should be understood as dynamic and context-dependent entities, only fully accounted by a process ontology. He follows Birch and Cobb's ecological model and Stein's proposal of enzymes as ecosystems. He adds that, like symbiosis in a termite colony, the behavior of molecular system is also defined by the integrated capacities of the system, capacities that emerge from within an integrated whole. Now we follow Fiorela.
Okay, so the final question is whether we can apply process ontology to drug receptors and biosignaling. So first, we can follow Stein and Guttinger's approach to account for the process of biosignaling. So let's remember the central points of them. It is the complex interaction with the surrounding water that ultimately shapes the way the enzyme behaves and that gives rise to the power to catalyze a chemical reaction. The two parts, maybe the enzyme and surrounding bulk of water, should be treated as a unit out of which the capacity to catalyze a chemical reaction arises. And also the interaction between water and enzyme is what brings about the capacity to catalyze the reaction. This is a picture of an integrated capacity of that system. So before we can answer the question, so let's take a look on how receptors and biosignaling actually work. G-protein coupled receptors, or GPCR as they are known, constitutes the largest family of cell surface receptors and the primary target of half or approximately half of the current prescribed and designed drugs. GPCR were initially considered to be relative simple signaling entities that conveyed information transfer in a unidirectional manner. Now it is known that GPCR behavior is highly complex. So they typically exist in a spontaneous equilibrium between two conformations, the active one and inactive one. that differ in their ability to activate downstream G proteins. G proteins are intracellular proteins connected to this system of signaling. In the native state, the receptor is maintained predominantly in the inactive conformation by intramolecular interactions within the transmembrane helical bundle. The spontaneous equilibrium heavily favors the inactive state. Agonist binding relieves these intramolecular constraints, allowing the receptor to relax into the active conformation, facilitating productive G-protein coupling. These constitute basically the extended ternary complex model, which is the model that represents the signaling process of this kind of receptors. Well, we have the signalsomes, so it is now appreciated that GPCRs do not solely signal through their titular associated G proteins, but through a series of additional signaling factors that possess a stronger and more stable interaction with the receptor. Primary among these additional signaling factors are arrestin proteins. The modulation of receptor expression, function, post-activation processing, and ligand selectivity is induced by stable protein-protein interaction of scaffolding or signal modulating factors. Such stable receptor entities have been termed signalsomes. This is a word to represent or to name the multi-protein complex that is involved in the process of signaling.
5 minutes, Fiore.
Okay, just in time. Here, so we present our concluding remarks. Of course, as Mariana said at the first slide, this is a work in progress. We still have to keep working to answer some questions, but also we wanted to present some concluding remarks. For example, the process ontology can or may be applied to receptors and biosignaling processes, especially if we consider biosignaling as an integrated capacity of these dynamic and the dynamic entities that are, for example, the signalsomes. Also, we still have to answer many questions, such as, for example, if this analysis can be applied to other kinds of signaling. So since GPCRs are not the ones that account for signaling process, overall signaling processes, or maybe other protein functions. Other question is, do protein dynamics actually ground on a process ontology? Or what about other kinds of molecules like supermolecular systems or in nanochemistry? Well, another concluding remark that we can claim that is this approach allows us to review the SS paradigm, since biological functions can no longer be grounded exclusively on molecular structure. And of course, as we have seen, relational and dynamics aspects are central to biological functions. So, well, as a work in progress, we are very grateful to hear your feedback and comments. So that's all for now. There you have our email addresses in case you want to know the references that are here. So, well, thank you all for being here.
Okay, thank you, Fiorella. First question, yes, Elena, please.
Thank you. Thanks for this interesting presentation. I just wish to make a comment because it seems to me that even based on the example that you presented, this review of the structure-function relationship dogma, as you call it, is already ongoing among biochemists and more in general among life scientists. I mean, textbook probably suffer from inertia because it's true that in textbook this issue is presented in still in a very dogmatic way. But the fact that the relationship between structure, function, and is much smoother than it was understood at the very beginning, I would say that's already well clear to most biochemists. I just mentioned a couple of examples. You mentioned the case of intrinsically disordered proteins, which are a typical case where you cannot ignore the role of the environment. And even the evolutionary perspective and the existence of convergent and divergent evolution is a sign that the relationship between structure and function is more complex than this dogmatic view is pretend. But as I say, I think that this is already part of the, let's say, of the cognitive baggage of most biochemists.
Sure, thank you, Elena, for your comment. We totally agree. So this is just one more argument to put in doubt the dogmatic vision, as you said, of this paradigm. So maybe dogma is a very strong word to refer to that. Maybe paradigm or model will be a more appropriate one. But of course, we agree that this is under discussion. By many years, not right now, but I mean a decade or at least 15 years or more like that. Thanks.
Jesus?
Okay, thank you for the interesting talk, Fiorella and Mariana. I have a clarificatory question. I don't understand very well how to identify the process, if it is in terms of the relations in which the molecule can take part or something like that. How to identify the process?
Can you repeat your question, because the audio was interrupted? Sorry.
So, no, don't worry. Can you hear me right now?
I guess so. Yes.
Can you hear me? Yeah.
Yes.
Yeah. Okay. My question is how to identify the process in terms of the relations or the molecules that participate in that process, or?
Well, that is something that we have to work out. We still have to work out. So I think that relations and I don't know how to call it, but the objective of the activities or the events that are taking place, that are becoming during time. I mean, not exclusively on the relation between other kinds of entities, but also on the purpose of something that we focus. For example, in this case, the signaling processes or the biosignaling process phenomena, cellular phenomena. I don't know.
Okay.
If Mariana has something to add.
Yeah, thank you.
So, Yaman, dear Yaman, is your turn now.
Yes. Well, I have. Well, first, maybe try to answer Jesus' questions. Maybe an interventionist account can try to help identify this process. Woodward has a very nice interventionist account to dissect the system and identify relevant components of the process. Maybe I'll send you literature, but my next question is, what is the difference between a mechanism and a process in your paper? And the second thing, moonlighting you mentioned for proteins in different environment. I don't think this is the regular case. Usually proteins don't moonlight. There are specific conditions. This is not the, but as far as I know, there was a last one, but I kind of forgot it. But yeah, I'll stick to these. I hear your comments on them.
Okay, thank you, Yaman. So yeah, I agree with you that moonlight proteins are not the usual case, but they have gained interest during the last years. But intrinsically, these other proteins are more common than moonlight proteins, for example. And well, we also mentioned the role of epigenetic processes or imprints that may influence the final shape or the final function of some macromolecular. Well, that I don't know if I answered that question. I know that they are not the usual case, but they have gained interest in the last years. Well, the other question on mechanisms, again, to be differentiated processes. Well, on the account of Dupré's process ontology, mechanism still grounds on a substantial ontology. So it depends on the fundamental entities that do the activities are the entities involved in the mechanism. So that what he says, and we try to insert on his line of thought, that things are precipitates of processes. So that is not to say that things that do not exist, but this position defends that the more fundamental entities are processes. So if we consider something as a thing in the classical substantial way, that is because we can consider it a thing in the time scale involved in the phenomena under study. I mean, for example, you have RNA, a messenger RNA molecule that exists or become to exist in a matter of minutes, for example, and then it is disintegrated or dissolves or something like that. I don't know the word, exactly word in Spanish, in English. So we can consider it a thing within the time scale on which this phenomena is under study and in connection with the other processes that are occurring simultaneously. It is a matter of time scales that we see things as things and not the very big picture that, according to him, it is a process world, at least in the living world. I don't know if I answered. I'm sorry for my English, it's not very good.
No, it's perfect. Thank you very much.
Okay, now Cherif, please.
Okay, Mariana, if you want to add something, you do interrupt me.
Yeah, thank you very much, Fiorela and Mariana, for a nice talk. You touched about a very important area that I have not even thought you're going to talk about. I thought you were going to talk about something completely different when I read superficially the title of your talk. I thought you were going to talk about QSAR, basically, the quantitative structure-activity relationship, which when you don't have a known mechanism, the drug designers often use that kind of approach in order to predict the properties of an unknown molecule, hunch analysis,
Sure.
the Hammett constants, all these things. So basically my question is, what is your thought about these kind of approaches of small molecule QSAR type studies? Thank you.
That is okay. Thank you, Sherry, for your comment. That is a very interesting question. I think our plan is to work out this approach in order to expand it to other kinds of molecules. Not only, I mean, why do macromolecules like enzymes or receptors should be treated especially different, and why not to apply this approach to other kinds of molecules, like, for example, synthesized molecules or synthesized drugs, designed drugs, as you said. So we have to work it out. We have to still work it out because our intention is not to criticize the QSAR approach. I know it. I mean, the mechanistic approach or that approach that you mentioned have shown to be very useful in drug synthesis and to account on the properties of novel drug discoveries or homologies to known drugs. But maybe I think that we can still make an effort to apply a process ontology approach, even in those cases. But that is work we have to do during the next time. But I am very aware of that kind of approaches and models, and well, it is a challenge. We have a lot of work to do if that is our intention.
Thank you.
Okay, Ute?
Yeah, thank you for your talk. Very briefly, I would be a little more cautious on dismissing the structure-function relationship. I mean, or to replace it to such an extent by processes. I think it still works. Let's take the example of partially disordered proteins. They are highly functional. They are functional in the signaling processes because they are not stable, and that is very important for signals. So, and also their structure, even the disordered structure, is determined by genetically. So they are not something disturbed. They are highly functional proteins. I mean, I'm not talking about the Alzheimer things, but the partially disordered proteins. And the second thing, I don't know why you invoked epigenetics. Epigenetics at all is not really something new. It is also the epigenetic marks are put to their places by factors that are genetically determined and removed by them also. So they are part of the vast machinery, which is indeed very complex of gene regulation, but they are not the primary movers of it. So I would be also very, very cautious to use them for your argument. You know, I don't know, I haven't heard exactly what you are talking, but I'm just saying that I would be cautious. Thank you.
Okay, thank you, Jutta. We will take into account your comment. Of course, epigenetic is not a new phenomenon. There is a lot of literature that talks about or in depth of this phenomenon. And of course, we are not saying that due to epigenetics, we are in the position to reject that paradigm because it's still very useful and because that is not what epigenetic claims indeed. But we still wanted to find some examples to at least make it make that dogma, as we call it, in a more weak or smooth version of it. No, that not in that deterministic and unidirect, strong deterministic way, but maybe in a more smooth way or weak way. I don't know if you understand my.
I understand you, but I just think you cannot get rid of this deterministic thing. They are simply there. And of course, it is not, the lock and key doesn't exist anymore. Everything has become more plastic and so on. But, sure. but still, enzymes are specific, DNA is specific. So we use it every day in every medicine, in every research in genetics and evolution, etc. So you cannot get rid of it. I mean, you have on the fringes, you have some other things which are more flexible and prone to disturbance, but one should really be very careful to determine something. And epigenetics doesn't belong there. I'm just telling you, it doesn't belong there.
Okay, thank you.
Okay, thank you. We ran out of time, so I would like to introduce.
There is a question from Michelle. So
okay, we can have it. Yes.
Yes, of
Michelle?
course.
No, it's okay. It was just a very naive question, and there are many questions it looks like that have not been answered. So it's okay. I forgo my questioning.
Okay, sorry,
No
Michelle.
problem.
Thank you. So
Thank you for your comments.
Thank you. Michelle, you can write us if you want to. You can send us an email if you like.
Perfect. So thank you, Mariana. Thank you, Fiorella.